see them all. I thought it would be good for the board to at least see, like, what is actually going on out there with earlier detection? Like, how is it being used now, and what is the kind of future of this that I and, you know, other people who do a lot of this is. And so that's what I kind of wanted to go over tonight. You're more than welcome to interrupt me with any questions, and ask me during this. This is intended to be very informative. And so right now, the early, you know, what are the methods for detection of breast cancer? I'm gonna read some of this because it might be hard to see, but it's exams, it's imaging, and that's about it. That's all we have right now. We have ways to tell if people can get additional imaging besides mammograms. Mammograms are improving. Mammograms are gonna change a lot more in the next few years as well. They're probably gonna change in Greensboro by 2024. There's gonna be the availability of something called contrast enhanced mammography, which is something that has a lot of promise, and it's probably gonna replace or at least do some of what MRI has done. And MRIs for breast cancer are recommended, and for, you know, just surveillance are recommended for women. And this contrast enhanced mammography, the problem with MRIs is that it is a scarce resource. There's only so much time on the magnet, so you can't, not everyone can go get an MRI, and then biopsies are a pain. So this contrast enhanced mammography has actually fewer false positives, meaning that it's maybe a little bit better of a test. So that's coming probably to Greensboro by 2024. So we're having more things coming in terms of early detection, but they don't. Many times these still don't in general find cancer at an earlier stage, and so for example, lobular cancers, you know, those are still, you know, especially if you've got C or D density mammogram, lobular cancers in general still aren't found till a later stage, and because they're not detected well by current imaging. So some of this is going to help that, but we're still are behind with all of this. And again, you know, there's up to 10% of breast cancer still found as a stage 4 cancer nowadays. So it's metastatic, it's other places upon presentation at this point, and that's what I think what we're all trying to prevent. There are actually ways to try to even reduce risk or prevent breast cancer that we can talk a little bit more about it as well. This thing, I'm having to do this different so it fits on the screen, so I'm doing it from my phone, so that's why it keeps going on, so I'll try to remember to hit it. So the liquid biopsy or a blood test for any cancer, you know, there's been tests for all colon cancer, breast cancer, ovarian cancer, pancreatic cancer. There are blood tests, but in general, those have not been shown to be high or elevated until the cancer is much more advanced. So it's been elusive to find for any cancer, breast cancer included, and in large part because what you might find in a blood test is you have to have a method to detect a very, very small amount of material. And that's been the problem up to this point. And so all of the grants that you see, if you look at them, we've got 2 PhDs reviewing them now because they're, you know, a lot more some of the basic science than what even some of the clinical people do. We have to look some of this stuff at this point because it's really cutting edge, so they've been helpful. But they really fall under a couple of different categories. The first is they're all different ways to try to detect cancer. And there's a bunch of different ways. If you look at all of them, they're all different ways. There's some very novel ways to try to find these cells. And then they're all looking for something different in the bloodstream. And that's what really almost all of the grants that we look at are about. And so the 3 things that people are looking for now, and I'll go at the end, I'll kind of tie all this together, are circulating tumor cells. And I have a picture of this next, which you have a solid tumor. Let me take a step back. For blood-borne cancers, you can find these things easier. There's a blood test, you can find them easier because it's in your bloodstream. These are solid tumors that are related to the bloodstream, and they release something into the bloodstream. So one thing they release are actual tumor cells. So they shed viable tumor cells into the bloodstream as either single cells or like a cluster of grapes, and they shed those in. And those are one of the things that these blood tests are trying to detect. There's also cell-free DNA. So dead tumor cells die just like anything else. If you operate on a tumor or you see it after you've given chemotherapy, you'll see dead tumor cells in this. Sometimes they outgrow their blood supply and they become necrotic. Sometimes you give them chemo and they die. And then you do surgery and you see evidence that there was cancer there, but they're dead cells. Those cells release DNA. And so that DNA has some specific mutations that are related to tumor. So they've got things on them that identify them as tumor-related DNA as opposed to our regular DNA. And you can detect that in the bloodstream. The other thing, and this one has a lot of promise, we've looked at several grants from these more in the last year or two than prior years because this has become a much hotter topic. And they're extracellular vesicles. Basically, they're like bubbles that contain stuff that the tumor cells release. And so your body does this for a lot of different things where it releases things in these little carriers or bubbles. They actually get released in your bloodstream and they have characteristics of your tumor cells. So all three of these things, all of these things we're doing are just different methods, different tests to find one of these three things basically. And so, it's kind of a crapshoot now to basically say, what test can we do to find this small amount of something in your bloodstream? And so that's the vast majority of what we've approved, and then funded, from this organization. And all of it is extraordinarily important. So here's a little bit of a picture here. So you'll see, hopefully this thing stays on while I do this. You see if this is a primary tumor here, they sit next to blood vessels and they release things into the bloodstream. The idea is, if these are tumor cells here, and they're viable tumor cells, if they eventually, sometimes they can implant somewhere else in your body, and that's would give you metastasis or what's called stage 4 disease. But all of these things are getting released here into your bloodstream. And the idea, whether it's this ultracentrifugation where you spin all of them down and find these different parts of your blood, or all these different assays, is that you do this and you find these things in your bloodstream. But that's the theory on how it works. The theory for breast cancer, for example, when this first started in the 1800s and early 1900s, it used to be thought you grew a cancer, eventually it went to your lymph nodes and underneath your arm, and then went other places. Well, that doesn't always happen. Sometimes it just goes directly in your bloodstream. And why do women who have a stage 1 breast cancer or stage 1 of any cancer, why do you develop stage 4 disease where it's disseminated throughout your body in the long term? Well, that's because there is this bloodstream route. That bloodstream route is now really what is going to eventually make this liquid biopsy to try to find some of this. The issues are now is how do you find the small amount of this stuff, and then what are you actually looking for? This is just another depiction of that, and you'll see, a cancer starts, it becomes invasive, it can release these cells, the little purple things you see in the bloodstream. Eventually, it sheds some of them. Some of them are never going to implant. They're just going to die. They're not going to implant somewhere. But for some reason, some of these go to the liver. They go to your bones. They go places. They implant, and then they start growing there. And so this is a lot of what I wanted to talk about. So how do we use it? So there are blood tests for breast cancer now that are being used, that are commercially available. None of them are for detection, though. Okay, not one of them is anyone goes to the lab, you go to LabCorp or wherever, you get a blood test and you find out you have cancer. But, and these have just come out since I think 2019, the first one was actually approved. And I was just up front, I'll be honest, none of us have any idea how to use these right now. But what they are is, and what they've proven is that, tumor DNA, if you what's what we call minimal residual disease detection, or MRD. So after you get treated for breast cancer, whether it's after surgery or after chemotherapy, most of these tests, what they do is they are tests on your tumor. So these companies take a piece of your tumor and get the makeup of it, and then draw blood on you to see if any of that tumor DNA is in your bloodstream. Okay, so right now at least, it's used for, I hesitate to say it, for monitoring, but we don't know what it means to actually monitor it. So right now, the idea that being able to detect this stuff after either surgery or after you get chemotherapy, or if you have stage 4 disease and you're trying to monitor, kind of how you are doing before you, start doing scans and everything. It is being used some for this. There's not any studies that say how to use it. It's all over the map right now, and the absolute numbers, no one knows really what to do with. But the ability to detect this DNA based on somebody's primary tumor does exist and is being done. And so, for example, it could measure your treatment response. It could, say I do surgery and you realize that their tumor DNA in their blood goes to 0 after that. Maybe at some point, maybe that person, in conjunction with the current stuff we do, maybe you don't give chemotherapy, or maybe if the current stuff says don't do chemotherapy, but if they still have tumor DNA in their bloodstream, maybe you're going to do that. That's where we kind of are, and those are the studies that are being done with this right now. Again, the problem is, say you have non-detectable DNA after treatment, then you follow it every 3 months and it's going up. Well, what does that mean? I don't know. What do? You start doing PET scans? We don't know the answers to those things. So this is really still in its infancy, but the technology does exist now with at least a couple commercially available assays to actually do this. So one of them is called the Signatera. This doesn't show up great on there, but it's commercially available from a company called Natera. Again, it's the patient's tumor that's used to develop this test. And what they're talking about, what they think can happen is about 20% of people with breast cancer get chemotherapy first now. Most people who have a lymph node positive, who have what's called a triple negative, meaning the estrogen, the progesterone, and what's called the HER2 are all negative, or if it's HER2 positive, are starting with chemotherapy at this point. And so one of the things that they're trying to do with this test is look at your response to neoadjuvant chemotherapy. So if you give chemo first and then you do surgery, there's a lot of benefit to that. Number one, the original reason why we did this was to try to reduce the rate of doing mastectomies, trying to convert someone from having a mastectomy to a lumpectomy. And so that still is true. But the other more important thing is when we give chemotherapy first, Ken gave someone chemotherapy, and then I go take it out. The pathologist looks at the actual response of the tumor. And so that response of the tumor is predictive in how they're going to do over the long term. If they have a great response, that's right. You're on the right track. You probably don't need to add anything else. If they don't have a great response, there's different things that get added on now that have been studied. So there's a lot of benefit. Are these circulating tumor cells or cell tumor DNA? That may be another way to look at this. The other way is just surveillance. Are you going to survey? Now, right now, once you have breast cancer and you get followed, it's mammograms once a year. It's an exam 2 or 3 times a year. It's some routine blood work. But that's all we're doing at this point. There's not any standard imaging. There's no standard CAT scans, bone scans, PET scans at all. And that's partly stage 1 and 2 breast cancers. It's just exceedingly rare to have a recurrence with the treatments that are now available, but there's never been anything shown that outside of waiting till there's really symptoms and then figuring out or a lab abnormality, that's better than doing anything else. And honestly, some of the treatments are pretty toxic that we do too, or a lot of surgery, a lot of radiation, so it's a balance. But these circulating tumor cells or cell-free DNA may end up, and that's what this company is trying to figure out. The other thing it can do is for metastatic disease. Metastatic disease, especially stage 4 disease, has gone to the bone now. That's not a biologically aggressive breast cancer, one that's estrogen receptor positive, for example. There's not even a lifespan you can give those people at this point, because I've got people who've been alive 10 years with that now, and 8-10 years with stage 4 disease, just because stuff develops, new treatments are available. And so it's one of those things, you're good at treating it, but you also need to give those people a quality of life as well. So the idea behind some of these tests are trying to figure out when you need to institute treatment, but also when you can back off a little bit too. Let me get to the next one here. So again, the current usage, there's another company called Invitae. They're also a genetics company. They've got one of these commercially available things that looks at your particular tumor. They are looking at, risk stratification of patients who have a higher risk of relapse after surgery, risk of recurrence after surgery, and who might benefit from chemotherapy. There are standard ways we already do this that have been validated and studied. This would just be an adjunct to that. And they've done some studies, and this again is still all really in its infancy as well. But they've looked at this and they've followed people before relapse, and it looks like this test finds it earlier than kind of the standard ways most of the time. What that means or what you do with that is what we're not sure about yet. And again, therapy response, it could be escalation of treatments. It could also be backing them off. If you've already had chemo and there's side effects again of all this stuff that we do, maybe you have a great response. Maybe you can actually back off and do less over the long term, which would be really good as well. And again, the surveillance part of this, but what we're looking at really is this going to help? But these companies both have figured out a way to actually do this test and get a number that tells if that's the case. The issue is, compared to what we're talking about with an earlier test, is that these are for a particular type of cancer. So the person has a type of cancer, it looks for that. Breast cancer really has kind of 4 main types, and there's a lot of heterogeneity, a lot of difference in each one of those groups. So a single blood test like you're doing for these, it's gonna, it might detect one type of cancer, but it's not gonna detect the vast majority of all of the breast cancers that are possible in that person at all. So that's the issue right now, is that these tests, the assay works, meaning the method that they use to find these work, but they're just looking for one thing. So where does that leave us? I was actually talking about this right now. So, and the other issue, earlier stage cancers, early stage 1 cancer or DCIS, a stage 0 cancer, there's going to be less in your bloodstream is what it looks like. So if you suppose you have a stage 3 cancer or stage 4 cancer, you're going to have more in your bloodstream. So there's fewer detectable markers. We have to be able to detect a very small volume, and it needs to be quick. And show, there's studies that show that this can be done, not really in breast cancer yet, but there's studies that show that for some other cancers, even some solid tumors, this can be done. It's just not available in breast cancer yet. And so what I think, honestly, the future of this is going to be is really what I write here is a multi-parametric assay from several sources. And what that means is, I think what it's going to mean is all of these different things we've looked at, all of these different methods to find these cells, it's probably going to be multiple methods, and it's probably going to be looking at potentially all 3 of those things that I have written down. Maybe it's cell-free DNA, it's extracellular vesicles, and circulating tumor cells. And so I think in the end, it's going to be several of these tests that get run through several different methods. And that's what's going to be the future of this. And why I bring that up primarily is, that makes what this organization does extraordinarily important, because all of these grants we have looked at are really looking at all of these different ways to detect all of these different things. And I was talking to Martha earlier, and we fund some of these, and some of them are negative. Like some of them, they're just not ever going to work. You didn't know that before you did the study, though. So, and this is true for anything that we do as physicians or researchers. A negative study is just as good as a positive study, and it's just as important as this. So, a lot of this funding we've done, these studies are negative. Well, either you did it wrong, or that's just not going to work, and you need to go down another route. So, some of these certainly have been negative, but it's as important when you're trying to figure this out and come up with something new, really, to figure out what does work, but also what does not work. And as an example of what does work, this is Vidarzi. They used to work for UCSF. Now they're doing a lot better. They work for this company that's out in Silicon Valley. And so the initial grant, was in 2017. It was when they were at UCSF, and it was leveraging these little cancer-specific small RNAs. The company now calls them ONC-RNAs, ONC RNAs, because they had to call it something fancier than that, for early detection of high-risk breast cancer. So they actually presented their results initially in 2021 at the San Antonio Breast Cancer Symposium. For some reason, biggest breast cancer conference is in San Antonio, but it's been that, I don't know why. I wish it wasn't there in December. Anyways, but that's where it is. But they presented this latest one in 2022 as a poster there, and this company XI has this now RNA-based liquid biopsy platform, and they're trying to work on a lot of different things. Part of what their grant was is they're combining the detection of this RNA with this machine learning system that they've developed to detect breast cancer across all stages and all tumor sizes. And they're reporting, at least in this initial data, that with the sensitivity and specificity. So it's a good test by these numbers is what that ends up being. So the test actually, that's a pretty good blood test right there for almost anything with those numbers. So their test actually works in detecting this RNA that is released from the bloodstream. RNA is one of those things that's really difficult to manage sometimes and deal with and test for, but they figured out a way to do a blood test and then find this. And right now, their next step is really trying to do some clinical trials with this. But that's a grant that the initial one in 17 that this organization funded that is now, being presented to a worldwide symposium, and looks like it actually has some promise at this point.
Nice.
Yes, and so again, that's a lot of what I talked about right there. And what else can be done? I think this organization is on the right track. I think part of why I want to do this is I wanted everyone else to feel like all the stuff that we're doing, it matters a lot. There's been some fruit that has been born already from it. There's other things I think we can do to try to prevent breast cancer earlier, but I think we're on the right track with this, and I appreciate all the work that all of you guys do, and let me still be a part of this. Thank you all very much. That's all.